Good. Right. So, yeah. Shall we start with the questions, comments? There have been fantastic talks over there. So, well, first one, just to get the microphone.
Yeah, one thing is that fantasy. Hello? So, for Francisco, so are there known repressor of the CRISPR system in bacteria? Like normal, like for example, you know, like any other thing in biology, you can be resistant to any biological process. Are known inhibitors of the CRISPR system in bacteria?
Yes. Okay, there are inhibitors of the CRISPR system in viruses, of course. Okay. So there are several viruses that have anti-Cas system. So in fact, recently it had been discovered, because we knew about that a few years ago, about systems that inhibit the type 1 systems, type 1 CRISPR-Cas systems. But of course, the ones are more interesting are those of type 2. And quite recently had been identified several anti-Cas proteins in viruses. So they are.
And my other question is like related to the evolution of the system, right? So it seems to be lost in mammalians, for example, right? In most mammals. But that could be, for example, of some utility to get rid of transposable element, right?
Sorry, the
whole CRISPR system could be somehow good for a mammalian genome to get rid of transposable element, right?
Yeah.
So what's your idea about like why is not present the CRISPR system in a mammalian genome, for example?
Yeah, that's the everlasting question. So the thing is, they probably had it initially, because in fact, as I mentioned before, they are present some CRISPR remnants in mitochondria. So at least they were in organella of eukaryotic cells, and probably they were there. And they were lost because eukaryotes invented something better for them than the CRISPR-Cas system, a system that eventually can take pieces of their own DNA and kill the cell. So this, for a pluricellular organism, is not a good business. But anyway, for yeast are unicellular, so they don't have a CRISPR-Cas system. And probably they had it initially, at least the ancestor of the eukaryotic cells. And they probably lost it because this CRISPR-Cas system, as an immune system against viruses, is not neither something especially important for yeast, because they are not frequently infected by viruses. So anyway, I think someone asked me a question related to this one, and we came to the conclusion that probably initially there was a very complex CRISPR-Cas system involving also Argonaute, that, as you probably know, is a protein of the RNA interference system of eukaryotes. So maybe it was a very complex system, also including Argonautes, and Cas1, Cas2, which are the ones involved in taking new pieces of DNA, were just removed, and the others, Cas 4 to 10, were replaced with other proteins that make the rice complex of the RNA eukaryotic system. So probably was replaced by this system. Yeah, it's just a guess. I love that.
I was reading the other day an article from a scientist called Aubrey de Grey. This person has been doing research on, well, he believes that we have the science and the technology to reverse to stop or at least reverse the most negative effects of getting to an old age. Okay? And he was talking about different areas where we can stop aging, like modifying cell mutations or getting rid of all the junk that accumulates in the organism when we are getting old age, and many other things. But my question is CRISPR one of those techniques that we can use to revert old age, and how close are we? He believes that we are very close to get some kind of immortality, or at least a very long life. Is it possible?
I don't think I'm the best to answer that question, because I am a specialist in organisms that are immortal. In fact, that is bacteria. So I know almost nothing about eukaryotes. So, you have any hint on that?
Well, if I don't know, I think that probably the context where you talk about immortality, it's really different if you talk about a body or if you talk about a cell type. So I think that for some cell type we are able to revert their aging phenotype, but talking about a human being and being mortal, I think that, yeah, that's quite of a science fiction yet.
Right, so I have a question actually about using CRISPR in animals. So it's being, it's basically for Professor. Some people is talking about not only using it in the existing animals, but bringing back to life to extinct animals, like the mammoths. What do you think about it?
It's a so not in the obvious form, not from the point of view that you could find a frozen mammoth and resurrect it with CRISPR per se. I mean, the argument there is, from a practical level, that getting that absolute intact DNA is challenging, and secondly, who would be the womb that carried the mammoth? But another point would be, let's take dogs. We have wonderful, diversity. I'm not a dog person. With all sorts of faces and sizes and shapes and attributes to them. Some are good hunters, some are very good at sitting in front of your fire, and all these different things. And they've all derived from the wolf. Every single one of them is derived from the wolf. And if you look at the genetics, yes, there's a lot of mutations between a Chihuahua and a wolf. You might believe that there's slightly less between the Alsatian and the wolf, and that's probably true. So you could, with CRISPR technology, you could go into a Chihuahua or a wolf and slowly change every mutation back to the wolf. So you could do that. Why? That's a hard. But so CRISPR per se is not going to bring back dead things. And even beyond that, most dead species, not all, but most dead species have died out for a reason, and that reason is probably still here. For some, it's either a lack of environment for them or a desire. We still have wolves in Scotland, and you may still have wolves in Spain. I don't know. We have places called Wolf Hill, and we know wolf was shot. Bringing back, well, I still have some wolves up at Kincraig, but so it's not a tool per se to bring back immortality or bring back the dead.
I just wanted to follow on from that because, as you know from our conversation just before, gene editing of animals is something that I have an ethical interest in. You said, why would you want to change a Chihuahua back into a wolf? Fair enough. But one of the reasons I think perhaps we need to look into gene editing for dogs more is that, dogs are our companion animals, well, for some of us anyway. We care about their welfare, we want them to have good lives, but at the same time, some of us want dogs that fit particular aesthetic or practical purposes. You might want a small dog because you live in a flat, or a dog that can rescue you from fires because you live far away and your house might burn down. So we've bred dogs to have all of these characteristics, but at the same time that breeding has come along with the accumulation of some deleterious mutations. So there is a whole list of all of the various problems that pedigree dogs can suffer from. Now, I believe that about 20% of these are thought or known to be caused by a single monogenic disorder. In other words, they'd be really tractable targets for gene editing. Now, you might say it's wrong of us as humans to want spotty Dalmatians and wolf-like German Shepherds and teacup-sized Chihuahuas, etc. But the fact is, if we're going to continue making them, and we are able at the same time to ensure that our Alsatians' hip sockets don't fall out, or our Chihuahuas don't die when they have puppies because their hips are too small. If we're going to continue making them, and we could make it so that they don't have these health problems, oughtn't we to do so? Isn't that something that we owe to our best friends? The other point that I wanted to make about the use of gene editing and conservation, I completely take on board what's just been said about you can't use it to bring back endangered species. But something I've been thinking about is, Bruce, you talked about using gene editing to either introduce alleles from parts of the non-breeding population or to fix the frequency of alleles that are otherwise rare. Now, one of the issues with conservation for animals that are down to very small numbers is lack of genetic diversity. For many of those animals, we have a picture of what their previous genetic diversity was because we have things like museum skins and so forth. Would we one day think about using gene editing to say, okay, this was the koala genome of 100 years ago, as we know by this collection of skins, the gene pool's much smaller today. Can we start putting some of that diversity from not the contemporary non-breeding population, but the historical population back in, and thus increase the genetic resilience of the species? And that's something that I'm quite interested in.
So we have a question over there.
Hi, I would like to first thank you all for this interesting talks we hear today. And I have a question more of the related with GMOs, so probably it's more related for Bruce. Speaking about GMOs with people, many people are against the idea, but not just for the GMO themselves, but for the use that the industry, the advantage that the industry takes from GMOs. For example, make the crops infertile and these things. And I just wanted to ask, in the case of the animals that are being created in the university, like for example the ones in Roslin, what happened with the patents or the who owns the right of those animals? And because they've been created in a public university, does it mean that they will be publicly available, so if a farmer can take it, or they will still be linked with industry and then maybe not available for everybody?
So there's two basic questions there. I'll answer the second one, but the one at the start which you didn't expand on, and that is we currently have industry. Forget about GMO, forget about editing. We currently have industry ruling our agriculture. Right? You know, you don't go into the forest and find the animal you want and pick it up. All the animals are owned by somebody. So there's no change conceptually from where we are today. We and the university any intellectual property that I produce. So the actual practical level that we are obliged by the university and by our funders is to attempt to maximise the return to the UK, basically, since I'm here in Scotland, for the stuff that we invent, if you want to call it that. So we, as inventors, on patents that we have, and we work with whichever industry or third party that we can to exploit that patent so that there's a return of value back into the university, which feeds on to research. Free of use, well, there's no of our economics that really benefits from that. There have been attempts at various ways to make things free of use, but basically then they carry no value, and therefore they're not going to give a return back to us. So if we had intellectual property free for anybody, then our taxpayers, which pay me and pay most of us in this room, would get no return. It would simply go to whichever company got there first. So by having a system, by having an intellectual property system, it brings you back into the research environment. Now, other people, I've got a nice jacket on, but it didn't cost me an awful lot of money. I bought. They're not very expensive shoes. I have not made personally a fortune out of this, but the university overall does make a relatively healthy return on its intellectual property, and that's a driver. So aspect of intellectual property that you've got to be aware of is that by patenting something, you put it into the public domain. Yes, there's a delay between when you file the patent to when it becomes public domain, but it's there. And if you want to, all the patents I have are over, apart from one, are over a year and a half old. You can go out there and find exactly what they say. So by doing so, you put it into the public domain. So I see intellectual property as actually a positive driver in the system. The return back, I think, is interesting, and different universities and different parts of universities deal with that in different ways. And we could have a big debate about which is the appropriate way to do that, but that's a separate issue. But I go back to what I said at the start. Whether a genetically engineered, a GM animal, or just a animal that we currently have, in most cases, that is owned by somebody somewhere, and there's no change.
So, I feel brave enough to ask for a comment of Dr. Chen, because I've noticed that people were creative, asking about immortality and things like that. So let's say that we would think about the future, and if we would edit our genes in such a successful way that our population would become a very healthy population of people, of humans, I think that there would be a problem of, we already struggle with the identity as humans, and that's about the population as Europeans or some people from some other places, but as humans within the other environment, and watching what we've done to animals as well and how we control their lives, I think that would be a question of identity as humans, if we would live in such a healthy way, and we would be even more dominating than we are already. I think that if we would be so healthy that we could control even more the environment, how to put it, so I think that we struggle also with the climate change, and if we would be so healthy that our population would grow even more than we are in the number as now. What's your comment on that? How could that go? What would we do next?
Okay,
that would be so dominating even more than we are now.
Thank you for that really rich question. There was a lot in there. I'm going to try and unpack. I saw at least three strands that I want to try and pick out there. So the first was, I'm going to start from the end because that's where I remember. The first is about dominating the environment and about our control over the world around us. I'm going to say that maybe that relates a little bit to this argument about proper agency and what should we be doing. I think when we think about exerting control over our environment, imagine if we exerted control in such a way that we could reduce climate change, that we could save the Great Barrier Reef, that we could stop the melting of the polar ice caps. I think we'd say, well, that's sure, it's exerting control, but maybe it's not a bad thing to do that. So control itself maybe is not the issue. Now, you might say, but those are reversing anthropogenic changes, so it's really just putting things back the way they were. But how far do we want to roll back? Which state of the world is the unspoiled natural one? Do we simply roll back to the pre-industrial era? Do we roll back completely to the pre-Anthropocene? Do we roll back even further to before the Great Oxygen Disaster, where those nasty oxygen-generating bacteria came along and made the atmosphere toxic for everyone? Which world is the world we should preserve and then say, okay, we give up control now? So I think in some ways, I wonder if the concern about agency when it comes to the environment is actually, it's not saying we shouldn't exercise it, it's our fears about whether we're going to exercise it in the right way and our uncertainty about whether we're actually going to act responsibly. But I don't think anyone's saying we shouldn't have control. They're saying, be really careful. You have control, you have stewardship of your world. Be careful how you look after it. Your second point was about if we keep on increasing the population, we're already at kind of tipping point. Is it going to be sustainable? What's going to happen? Yeah, if everybody lives to 90 in good health, the population's going to grow. If 3 out of 5 children in sub-Saharan Africa don't die before they reach 5, the population's going to grow. Nobody's making that an argument to say don't save children. Nobody's making that an argument to say don't make sick people well again. Yeah, the population will grow. At some point we'll have to start saying if people are going to live for a long, long time, we have to stop having children. We have to stop having children until later in our lives, or else the population will continue to grow. But you know, that's already happened. People now live longer than they did 100 years ago, and nobody has said, well, let's go around killing off the old people to make more space. Okay, so dominance, sustainability. There was one more, but I am not sure.
I forgot as well.
Sorry?
I forgot as
Okay.
well.
I hope that it was a great question. I'm sorry I didn't remember all of it to answer it as fully as I
Thank
could.
you for that.
So my next question, for example, is just for the researchers in the lab. So they were talking, so how good CRISPR is, how much we can do with that. So what's the limitations we have with the technique? So let's talk about the bad part of it. So what we would like to do, we cannot do with the CRISPR yet.
Oh well, so I think I'm not a genome editing person, but I think that probably the challenge with the genome editing that people are facing maybe still is on the off-target thing, even if now this one seems to be quite alright, I would say. For us, like people using the dead Cas9, I think that the challenge that we face is that probably we don't know how to control the system so much. So, for example, what I've been doing is that there are still things which we don't really understand, so why one guide is more effective than the other one. So there is much more to know about the control of probably like the efficiency of them on the genome. But yeah, I think that for what we do now, that is like a good point. For the genome editing, I'll pass it over to Bruce.
I can make a couple there. If we think what genome editing does, it creates a mutation in the DNA. And we've been creating mutations in DNA for decades. What it allows us to do it now is just much more effectively, much more precisely, and much easier and cheaper. So if you want from an academic, practical limitation, it's what we imagine we can do. All right? Because we can do it, but we have to be clever enough to know what to do. So, so that, you can like the answer or not, it's up to you. The point I get, which we're thinking about, is at the moment it's either a transfection, it's a chemical and an electrical impulse that you introduce the editor into a cell. In our case, we inject it through a glass needle, and there are some semi-high throughput mechanisms using transfection which will allow you to introduce editing, or the guides at least, into many cells and so forth. So that it's not the tool, diversity of the tool, which we're tweaking away at. The real challenges, or you could say it's an opportunity. A real challenge would be: can you actually envisage a system whereby you could deliver the tool easily across a massive space? Now that could be geographical, it could be time. Can you do that? So the example to say that it's even slightly possible is we've been hearing about these small plastic microbeads which are in our washing up for the clothes, which are covered. Imagine if they were all covered with editing tools.
Hi. Yes, on top, back, on the back.
No, I want to make a question.
All right, sorry.
You have the cue. You got control.
Yeah, this question is for Whitelaw, Professor Whitelaw. You said that the most efficient species you're working on is pigs and sheep, and you said that on them there is very little variability. Is that the only path you are following? And would you have any plan to keep or increase diversity?
So, I think it across clearly within a variation within our bred domestic animals. But the diversity is somewhat different to some of our rare breeds. And colour would be a simple example of that. We do have variation with colour in some of our animals, but we don't have the extent of colour that's in some rare breeds. So there is still diversity. The second question to do, what are we doing about it? So I was talking a few minutes ago that editing could be used to reduce inbreeding. If you understand what the genetic variation that is being inbred, which is a deleterious trait, as our royal families all know, you could actually use editing to remove or change that variation. So you could do that. Is that a widespread effect? Not in the short term, at least. Or not increasing diversity, but maintaining that diversity is through the large-scale genomic efforts that are going on at Roslin and elsewhere. So when we talk about animals in the breeding environment, we're selecting for that good trait that people want, will ensure that the amount of inbreeding or deleterious traits are kept to a minimum and maintain that diversity. The point I'm trying to get across is if that diversity doesn't exist in the population, then editing is the way to get it in there. But there's still variation that exists. Yeah.
So now in the back.
She was waiting for quite a while.
All right. One first, then the other.
It's more for Sarah. So you said that society now is okay with genome editing of different species, but in fact there is quite a lot of discussion about the use of GMOs for food industry, for using it in crops, genome escape. So I think that people are quite concerned about genome editing in different species and it affecting the common gene pool of that species. And it is normal, I think, that to have that concern also for humans. And also, we don't know the effects of CRISPR in eukaryotic animals, well, in animals, long term. So it should also be important to check the effects of CRISPR long term in animals and later in humans.
Yeah, definitely understanding the full consequences is important, whether we're talking about animal use, plant use, or human use. When I said that we are generally accepting of genome editing in other species, that I didn't mean to imply that we blanket accept everything. We are more concerned about certain uses, and so you brought up crops and food animals. But the point is that we're not objecting there just because it's a use of genome editing in animals in general. We're saying these particular species have a particular significance or are of particular concern. In the case of food, one might argue it's partly due to safety. I will point out that, okay, you say we don't know the full effects, we have to check what those are going to be. That's true, but we know, for example, the full effects of vitamin A deficiency in children, and we know that if we make a change and we put vitamin A in their crops, that can fix the issues with blindness. So this is golden rice, which was produced not by gene editing, but by other means. So again, it's that trade-off between what are we giving up if we don't do it, versus what risks might we be incurring if we do it. The risk question is one thing, but thanks for bringing up food, because it actually relates to something that I've just been thinking. It was prompted by this idea that, hey, we could edit white pig, what are they called? Big white pigs, large white pigs to taste like Blackfoot Iberico pigs. And I was thinking, food, when we eat food, when we consume food, we're not just consuming proteins, fats, carbohydrates, flavonoids, carotenoids, DNA. We're also consuming culture. We're consuming a story. I remember once having a meal that had been cooked entirely at home by a host family I was staying with, from vegetables they'd grown entirely on their property, and it was wine from their own grapes that they'd trodden with their own feet. Now, they could have gone down to the supermarket and bought all of these things, but the culture that I consumed, the story that I consumed was important along with it. And so when I go to the supermarket and I buy the real jamón ibérico versus the large white engineered ibérico, they might taste exactly the same in my mouth, but in my head maybe they're going to be different. Is that an argument not to make white ibérico tasting pigs? Well, it's not an argument not to make them, but we have to accept that maybe people will treat them differently. Maybe people will prefer the one that comes with the culture that they also want to consume. And maybe people will say, look, I can buy this one at a fraction of the price of the other, tastes just as good, it's fine. But that's a choice that should be open to them to make.
We have an example of that today with salmon. So we have a real example where we have two types of salmon. We have wild salmon and we have farm salmon, and we as a society cope with that and make choices on taste, on cost, on culture. So that type of discussion we as a society, I think, are very good at dealing with. We'll have to edit the throwing gene.
Thanks. So I'll try and make this quick. I work on genome editing in somatic tissues in vivo, which is traditionally done by gene replacement. Now with CRISPR we're trying gene editing, and it's very difficult in this kind of system with a diverse cell types to knock in changes into the genome. And obviously it will be a lot easier to do it ex vivo, which they've done already, and that goes back to doing it in the germline. So I guess my question is there a way to make this, is there other ways to make this a lot easier, say by making fusions with with either Cas9 or Cas1, Cas2, which is the type 1 Cas system, so that you can easily insert the genes without having to rely on the endogenous repair machinery, and whether it's worth doing this at all to begin with, or if it's going to become commonplace to do it in the germline before we can even get to the point where we can do this somatically in an easy way. I guess it's sort of a general question for anyone who wants to answer.
I would say it's if we wait till you can, and people I have imagined a number of diseases, somatic diseases, which we could treat using with editing technology. If we as a society were, and if that's important and justified, if we were to, as a society, to wait until we could do all that through the germline, then we'd be doing our fellow citizens an injustice. Whether we should try and modify the tools we've currently got, well, I think Francisco might be able to comment on that, but I would say yes.
I mean, there are many alternatives to the tool we are using right now, like as I mentioned before. So it's probably we will get some tools, I'm pretty sure about that are better than the ones we're using now in the very near future. I'm convinced about that. It's not just mutants from the ones we know, but probably different analogs that are present in nature. And I think you saw probably the image of just the analysis of a metagenome from one place, a particular place, and we found two different proteins that are not related to Cas9, CPF1 or whatever. Just in one analysis of a natural sample, we have obtained two alternatives to Cas9. So I'm pretty sure you will get something much better and more precise and more appropriate to particular applications and particular cell lines. And that's the possibility that you can get them. And also, I think if the question is we should improve the tools they are using now. Of course, they have to, and they will. For sure, you will get something much better than this. Then one question I still don't understand. Probably you know that the Cas9 derivative mutant that has no tolerance at all to mismatches. I don't know about any other paper after this one that use it. It's quite weird, because in fact it's just. It should solve all these off-target problems, but something.
So there is quite a lot of research going into that, Francisco, but the evidence coming out now is not as encouraging as.
Okay.
We'll wait and see. And dramatic, and that is your offspring, but if you're that's got the disease, germline's not quite so useful. Can I ask a question? I would like to ask Sarah a question, right? So, part of the consequence of drawing the line is that you can get a consensus around that line, and a big thing, you may know where I'm going with this, and that consensus is a dialogue between the line drawer and the rest of society. 14 days, 28 days. We've spent a lot of effort as a society around the 14 days. There is this pact between the regulatory system, the scientists, the industry, and society around, and now we're saying, well, we can actually go further than this, and the reason we've got is because we can. I'm not 28 day, but how does that pact bit between the line drawers and society, it carries a momentum of its own?
Thanks, Bruce. I think you're absolutely right. So the 14-day, 28-day thing is referring, of course, to the use of human embryos and the point after which you cannot perform research on or indeed grow them in culture. I think this really touches on the point that I came to on my last slide, that regulation purports to draw lines, but its purpose in doing so is not to define, here's the side of the line that's morally a-okay and the other side of the line is absolutely not okay. We need to have a line for a number of reasons. We need to have a line because scientists need certainty within which to work. We need to have some sort of agreement, whether it's in the form of a line or something else, as to how we, the various publics, feel about what's happening with science and the arena in which it's operating. So not everything is a bright line. 14 days is a bright line, but what you can and can't do prior to 14 days, well, that's something for which we have an advisory body that decides that, and we trust that body, we task that body with making good decisions that somehow balance out the interests of publics, the interests of patients, what we can discover through science, and the fact that all of those are constantly changing. So I guess, if anything, this sort of reinforces what I was saying, that when we think about regulating gene editing, or indeed any area of science, we shouldn't just be thinking about what's absolutely morally permissible and what's not. We should be thinking, how does the regulatory environment we have, how does the solution that we might be proposing, or the particular area around which the discourse is being conducted, how does that promote that pact that we have between science, society, publics, stakeholders, so that we can advance in a way that satisfies all of those interests. I don't know if that answered the question. I think more I was agreeing with you in a very long-winded way. Sorry.
The other aspect I feel is that when we draw a line, it's a temporary line. It will change. It may change 2 weeks down the road, it may be 30 years down the road. But it will change as we as a society grapple with the question in advance.
Are the lines the same for all around the world, or is only for specific countries?
So that this is opening up an entirely different, but a very relevant problem. At the moment, no, the lines are not the same all around the world, whether it be for embryo research or for gene editing. Should they all be the same? Now, if they're not all the same, of course, you get the problem of scientists say, well, I'll just go and do my work elsewhere. We saw this debated with embryo research, where countries that were contemplating a more restrictive approach, Australia was one of these, I was working there at the time, saw lots of scientists threaten to go to Singapore. Well, we'll just take our science and our business and all of the economic and scientific benefits somewhere else. So you have that problem. Scientific tourism also, I think, has more that, there's the impact, sure, on the country where the scientists are leaving from, but I think there's also the impact on the countries that they're going to. Just a few days ago, less than a week ago, John Zhang published his paper, finally, on the first so-called three-parent baby, the mitochondrial transplant that took place in Mexico. He's a US scientist. The family that he performed the procedure for were from Jordan. They went to Mexico to do it, and Zhang was quoted as saying, there are no rules in Mexico. What have been the consequences of this? So, okay, the US don't yet allow this procedure, not possible where the couple came from, so they went to Mexico. What have been the consequences? Well, and I owe this discussion as well to two of my colleagues who work on this, but in an area that's already hotly debated, the fact that a scientist has blazed in and done this very controversial technique has just lent ammunition to those people who would seek to shut down the legislation in Mexico. It's already a conservative country where things like women's reproductive rights and the freedom of scientists to do research is precarious. So this, if anything, has made their situation worse. Moreover, if you look at the reaction around the world, people have been saying, well, this is a problem of countries that have insufficient regulation and poor ethical standards, so there's a judgment problem going on here as well. A parallel situation, I think, has maybe occurred a little bit with the response to the first two human genome editing embryo studies being from China. I think there has been a little bit of a Wild East flavour about the responses of scientists in the US, UK and Europe saying, don't know what's going on over there in China, but it's bound to be dodgy. I think there's definitely a little bit of scientific/ethical imperialism in our attitude towards research that's going on in other countries. And so I think lines aren't going to be the same everywhere, but what we do in that situation of the lines not being the same, of a pluralistic global regulatory environment for science, can have really big implications, and I think we need to think carefully about that.
There's a question over here.
Okay, so actually my question was a little bit on that direction. So the thing that I was thinking is so who is really right to make these policies? Scientists, science policy makers, governments, and all that, because we are talking about embryos, and maybe in China they make these embryos and it's there, that's fine. But there are other applications that CRISPR can have, like these mosquito CRISPR edited mosquitoes that they say to get rid of malaria, or maybe now that it's trendy, the Zika virus. So that affects. So there are no frontiers for that. So if a country makes a decision according to their regulation on that matter, but affects the whole world or an ecosystem and things like that, who is going to really argue on that matter?
I think you've just presented a very good argument to say why regulation of these technologies cannot be the province of any one country alone, but it's got to be something on which we have a concerted global strategy. That doesn't mean that we need complete harmonisation. I'm not convinced harmonisation is always what we want, but we have to think about it as a global problem. I think you have made an excellent argument for that.
One thing that you were saying before is, or maybe I got it wrong, but it's like, well, if there is a benefit, why not doing it? I don't know if it was you or Bruce who said that. But it might have a benefit, but it might have horrible consequences on other parts. Like, for example, if we want to get rid of malaria,
so
fair enough. But if we get rid of mosquitoes, maybe we get rid of the ecosystem, the plants, the in a certain country, the food that those people take, and so this is the butterfly.
All right,
right?
I didn't mean to imply that every time there's a benefit you ought to do it. I misspoke if that is what you understood. I was trying to present I was trying to present a criticism of the argument that if there's any risk, we shouldn't do it, because everything involves some level of risk. But I certainly didn't mean to say, well, if there's any benefit, we should always do it. What I was trying to get across is that we need to balance the risks and the benefits, both of doing something, but also of not doing something. In other words, when we say, okay, there's a risk involved in gene editing mosquitoes, that doesn't mean we should never do it, but we have to think about what is the benefit of doing it, what would we be giving up if we didn't do it. Does that make sense? So for either option, to do it, what are the benefits, what are the risks? To not do it, what are the benefits, what are the opportunity costs?
My question in that sense is, so if a country makes a decision to follow up with that, but the consequences affect other countries, so shouldn't we have like a global way of...
Yeah, so that goes back to my first answer,
who should do that?
That we absolutely need some sort of regulatory strategy that goes beyond the national level, because science knows no borders and neither do mosquitoes.
Again, we're not picking on mosquitoes, but let's pick on mosquitoes. You've got to compare to what we do already, and many people might think the widespread spreading of DDT some years ago was not necessarily the best thing to do, but we did it as a society. We felt it was a really so again, not see that as an isolation. It's of what we can do, and we need to do that risk balance. So the first question we have to ask is, do we want to get rid of malaria? Is that important enough to actually target? And if we think it is, then we look at the options, and each of them, we have the risk and balance of those, and we may come to the conclusion that malaria is a really, really bad thing, but we don't have anything which we're comfortable using. Or we come to the conclusion that, we do, and actually this is the safest, but not absolute safe. And the idea that as a society we only go with things that have no risk is nonsense. And all you will engage with, you will jump into a car after you finish this, or a bus, and not think twice about it. And yet that's a very, very dangerous way to travel.
I have a question for Professor Mójica, and it's regarding finding out CRISPR as the immune system of bacteria, so brings the idea, right? Okay, we work on that, we get rid of bacteria, all this resistance to antibiotics and all stuff. But what if somebody thinks, well, this is a very good thing to make superbugs and to treat everybody with the superbugs? So how close we are to being aware that this is a real threatening, let's say?
Superbugs to kill people, you mean? Yes, of course. That has been possible for many years. You have the toxin produced by Clostridium botulinum, the botulism toxin, that is the strongest and more efficient drug or molecule to kill people. And we knew that for a long time ago, and now we are dealing with a CRISPR-Cas system that allows for genome editing, but we have restriction enzymes that since in the 70s have been using to introduce new information quite easily in bugs. So it was really possible to do this high killers a long time ago. So it's not now easier, in fact, to do that in prokaryotes. We have not gained much with CRISPR-Cas technology in prokaryotes. We have gained a lot in eukaryotic cells. So the situation has not changed really, but not in the negative aspect, but in the positive it is, because we know that bacteria have an immune system that can be programmed for good. To improve, to make them more resistant to infection by viruses and things like that, or interfere with antibiotic resistance transfer, or to generate these antimicrobials. So now we have a better tool, not negative aspects as far as I can guess, at least in prokaryotes.
Good. So we have, all right, several questions here, so starting there.
Hiya. My question is for Professor Whitelaw. Seems like now the concept of food security is justifying a lot of research and investing money in genetics and so on. So we know the advantages that we have with genetics, improving livestock and agriculture maybe, but we already know as well that there is an impact, a pressure, a social pressure, and environmental pressure, a cost effect, a cost cost-benefit effect on this pressure to the environment and so on. So my question is, are we going in the good path to alleviate hunger in the world?
So again, quite a complex question there. Good path. Well, the one thing about society is we all have our own thoughts and our own balances and our own lines that we draw, and they change over time. And the diversity that we have in our food system for some people is important, and other people it's not, and we as individuals choose which part of that food system we want. As a society that wants that diversity, then many of the approaches that editing could offer in plants and animals has an opportunity. I don't give the impression at all that it is the solution. It certainly is not the solution. It is one of the solutions we have to explore, and there are many other aspects to food. And if we could reduce the transport footprint that is associated with food supply, that could have a massive undertaking. If we could reduce the waste that we currently have in our food system, that would have a massive undertaking, and nothing to do with any high technology. So look at all of those and come together. If we rely on just one, I think we have a risk. That's all I'm trying to say. So, but I really like diversity. The mere fact that I'm speaking to a largely Spanish group in Scotland suggests that deep down you have diversity too. So it's one of the strengths of our society, and I'd go the idea, could we bring back animals from the past? You know, personally, I would love to see some of these, and we're excited when we hear that the Tasmanian tiger might have been seen. You know, we deep down think, well, that'd be great, but we realistically know it's about as likely as finding Nessie in Loch Ness. So, so diversity is good in my book.
Okay, it's more like a comment rather than a question, and it's basically what we've been talking about. What you've been talking about drawing the line in regard to plant sciences, and because I can think of, CRISPR-Cas9 or CRISPR technology has been widely adopted by plant scientists and the plant science community, but one of the challenges would be the regulation, because when you're applying this technology and you get a variety that you want to release potentially, it's been genetically manipulated, and even if you don't have the Cas9 or anything there, it should be labeled as GMO. But I also think that we have other varieties, wheat varieties or barley varieties actually growing in the field that have been released, that have been developed by other mutagenesis or by chemical mutagenesis, and that's accepted, and that's
so should we to paraphrase it, should be labeled mutagenized food, and have to risk, and if we don't attach labeling to risk, we run out of labeling for everything, which is chaotic. I ask, generally, when you go to the shop, supermarket, whatever it is, how often do you look at the labels? Picked up, and if no sugar is in it or salt, I would suggest that we just do that. I'm not saying that makes it right, but I'm just saying we have to be careful with labeling, and I think it should be attached to risk. And if it's risk, then we have to define what the risk is to do with that GMO editing event.
Look, I think we do want to know about risk, but I don't think labelling is all risk, especially when it comes to food. There's this trend to, when you buy organic eggs from local producers, they'll write on the box like which hen they came from, and that I don't think is about risk. That's not about thinking, well, Hetty gave me a funny tummy last time, so I'll have eggs from Sadie this time. That's about people liking to know the story of their food, and that's not really about risk.
That's marketing. That's simple.
It is marketing. I think so. The problem comes when we can't tell whether it's marketing or risk, and so people have this idea that, okay, I'm a bit skeptical about GMO, so the marketing thing is let's label it GMO free, and then that reinforces the idea that GMO is a risk label and it shouldn't be. So I think there's that, what are we even labeling about? I want to say I feel maybe this drawing the line metaphor has been taken a little bit too seriously. I tried to indicate my skepticism with the whole idea of drawing a line, and to show that I think it's not about bright lines so much as it is about finding a field for discourse. But I certainly don't think that gene editing is the line and everything before that, selective breeding, chemical mutagens, etc., is fine, and then gene editing we need a line so people know not to go beyond that. I don't think that at all.
A couple of quick comments as well on that. So we've had a lot of discussion about the risk benefit and how what we can do with this, and how which applications we can go for. Remember that what you've got to really think about is that this is a revolution. This is a revolution that's going to have an impact. I've got no doubt about that. But it's a strange revolution. It's not that we've changed our allegiance to something. It's not like the Industrial Revolution. We didn't have engines, we had engines. It's not that. It's not like the space race where we hadn't been to the moon, we are being to the moon. This allows everything we've done before, but to be done easier, quicker, and by more people. And it's that more people bit which is the revolution, which is a different way of thinking about it.
But we're just finishing. So we have just a question, and I've seen you just waiting for the question. So I'm going to allow this last question. We have just some time now for having networking. They will be around, so plenty of time just to have some discussion with them. I'm sorry for cutting you guys.
So I still have the right to the question.
Yeah.
Yeah. Okay.
You have your
Yeah. This is well, of course, it's again about drawing lines. So this is for Sarah, of course, and this is about also enhancement of human beings. And well, I believe that our society, our so-called democratic society, is the unfair by default. I believe that we need social classes, and we need poor people, we need rich people, and I don't believe in this society, but I think our society is like that. And from this point of view, well, nowadays our society has different tools in order to secure the society itself, like repression, unemployment, war. Don't you believe that if we go too far in drawing lines, we don't draw a line concerning enhancement of human being, this will be another tool in order to secure our unfair society. Nowadays, there is repression, but still people can complain, still people can revolt. What will happen if we got like selected individuals that are much better than the rest, and then we have a class of, well, people who are not able to think about themselves and everything. I don't know anything about genetics. Probably I'm going too far, and probably this is just the argument in the book by Huxley that you mentioned in your talk. But what do you think about that?
So the question is, could gene editing and genetic enhancement become a tool to increase inequity? Yes, absolutely it could. So can cars, so can computers, so can a lot of other things. This is not a reason not to have any of those, but it is a reason to think about how we're distributing benefits, not just the potential benefits of gene editing, how are we distributing benefits at the moment? Should we be doing more? The problem in an equal society isn't down to any specific technology. you're right, it is a problem now. That also means it's going to continue to be a problem whether or not we introduce gene editing, whether or not we allow enhancement. It's going to continue to be a problem unless we do something now to fix it. So it's not really a question about enhancement or gene editing at all. This is a question about what's broken in society today and how can we fix it, and I wish I knew the answer.
All right, so thank you very much for this. I think we have just to, the last thing we have to do is just closing the session, but before that, we have to, well, announce the best prize. So for that, I would like to ask the Vice President, Dr. Estrella Luna, to help me with the good news.
Sorry, I didn't even remember I had to do this.
All right.
Yeah.
Okay, so thank you very much to the panel for deciding that Eugenia SRUK is the winner. So can you please come? Then, all right, so we have 3 travel awards for the people who came just from different parts of the UK, and they were members of the society, and they brought a poster. So we have, let's see,
Isabel Romero is here, yeah. And David López Martínez. And we had as well Jacobo Elias, but he had to go. But all right, so we have another.
To finish the day, I would really like to thank the speakers. It's been such an amazing day. We learned loads of things. I think that everybody's been just talking about how good it was today. So what we've done, we have just a very little thing for you guys. So we have something for Professor Mójica, which is a little bit of the Scottish culture, and it's not a kilt. And this is just a bag from my inspector, that she's also scientist. I took it for us. So thank you very much. I would like to ask you a bit to give something just Spanish for Professor Bruce Whitelaw. For Antonella. Well, it's all right. It's also Spanish as well. Thank you. Working over there, because we really want to thank you your effort today to be the host here and for all your help during the time. So right. Just the last thing I have to say is, well, closing the symposium. It's been really nice. So hopefully, all of you have enjoyed. Thanks again to speakers. Thanks to all everybody just to participate, to make the roundtable really. And it was a real shame just to have to stop the discussion. But it's like time is going over. So just finally, well, obviously thanking the representation of the Spanish Embassy here, Lorenzo, Fernando, our sponsors, the Fundación Ramón Areces, OXA, CETID, VWR, and CGEM for their support today. And I would really like to thank especially the working group of the SRUK Scotland, as well as Javier and Giovanni, because all of you guys, it's like all the passion, the effort you've been doing all this time is what it makes today possible. So thank you so much. So on behalf of SRUK Scotland, I would like to thank to all of you, and we hope you enjoy the day, and we hope to see you again on another meeting. So now there's some drinks over there, so let's go and enjoy.